thanks very much. It's really a honor to present here. I thank the organizers for inviting me. This is new to me. Like I don't see patients, so I take this opportunity to tell that we are in a much better shape than we are 5 years ago. I'm an optimistic person. I want to start with there's really hope. This is a title that I didn't choose, Dennis chose. Why TEAD is a convincing target for EHE treatment. Like David said, I mean, I've been working on TEAD for 10 years. So this is really a good stage to present like why TEAD is a convincing target. By the way, like I mentioned, I'm Dr. Pobbati and I work in Rubin Lab. And I'm also an assistant professor at Cleveland Clinic Lerner College of Medicine. So this is how EHE looks, the genetics of the EHE looks like. So basically, it's a disease which a fairly clean background. And what is shown here is the chromosome profile. And what basically is causing EHE is a chromosome mismatch. And if you look at it, basically the arms of 1 and 3 have been mismatched. I mean, I'm not a cytogeneticist. I have to really look at the arrow to see what's really happening, but I'll make your life easier by saying this. Imagine this to be a chromosome 1 and 3. And what has really happened is that it has just changed arms. So there's a chromosome mismatch. And what scientists call this as chromosomal translocation, what we call is not important, is basically is a chromosome mismatch. So why is this chromosome mismatch important? Because this mismatch has created a monster chimera, which people have all talked about in a couple of talks. And this monster chimera is called the TAS-CAMTA1, or TC in short. So what does it do? It actually couples two monster properties. You can kind of think about like a minotaur. So it has the body of a human, it can think like a human, but it's as powerful as a bull. So two things come together and you create this monster chimera. So part of that thing, the ability to turn on genes, it comes from the TAS part. The other monster part, ability to stay in the nucleus, that comes from the TAS-CAMTA1 part. So these two properties combined together make this monster. So this monster has to stay in the nucleus, and it does this function like previous talks they mentioned. Basically what it does, it turns on genes. So it acts as a master switch to turn on genes. So I believe people are there from all from all over the world. So in the US, this is on, whereas in the rest of the world, this is off. So basically, I'm talking about a switch which is on. So basically what it does, it turns on genes. I purposefully showed genes as a dot, two colors. Some of them are green and some of them are red. So there are some bad genes that we know of. Whether it really causes the cancer, we don't really know for sure. One good example of the bad gene is CTGF, Dr. Rubin mentioned. So basically the end effect is this chromosomal mismatch creates a monster. And how does this EHE compares with other cancer? Again, the dots are genes. So when you look at it with EHE, it's a rather simplistic view of it. So what you all see is just this monster chimera. Whereas other cancers, they're not as fortunate because there are several of these which could be bad guys, and we really don't know whether this is causing cancer or not. Whereas in EHE, we are fairly sure that because there is a clean background, this monster chimera is probably the one that is causing the cancer. So that is not proof enough. Just because it's the only consistent abnormality in there, this wrong timing, doesn't mean that, are you able, I need to shut this thing out. Are you seeing this thing that I'm seeing? Continue with free version? Probably not.
Right now, we're seeing the slide that says, does TAS-CAMTA cause EHE with the liver in the middle?
Okay. So you're not seeing what I'm seeing. That's good. So, all right. So you have to prove that the monster chimera actually causes the disease. So you got to give it to Dr. Rubin. I mean, for carrying out this, like he's like an emperor. He has this great plans to really prove that this monster chimera is actually causing the disease. So basically, it's not enough to say that this is the only abnormalities that is present and that causes the disease. No, you have to show it. And this has been elegantly shown in this using a mouse model. And this mouse model basically has only that monster chimera that is expressed. And, and as described in his talk, we do get EHE, and he described in really all detail thoroughly with all histology and any other studies that shows that this is really an EHE. Of course, the emperor has his plans, but you need a general to execute this plan. And that general is Caleb Seavey, who executed the plan. So now we have a monster chimera. And you would ask, okay, this is a monster chimera is there. Why don't you target that monster? But at this stage, I want to take a step back. To slay monsters, what weapons we have? So weapon one is a small molecule. Basically, I did it on purpose. This is a small molecule, Gleevec. It slays another monster. It's one of the most successful targeted therapy drug. So you could, in principle, think about using weapon one, which is a small molecule. Or in other words, these are the drugs that you take orally. And this can penetrate the cells. As I said, this monster resides in the nucleus. It can go into the nucleus and attack its monster. But is that the only weapon that we have in our arsenal? No, there is another weapon, which is called as the antibody. This is the body produces like to fight infections. But the prerequisite for using this weapon is that we need to know a bad guy that is sitting on the surface of the cancer cells. And this guy has to cause cancer. And we don't know who that bad guy is in EHE. Another thing that Dr. Wilkie already touched upon is immunotherapy. Basically, can we use or leverage our own immune system to fight the cancer? So far, we don't have enough data to say that this works, but if there is enough research to show that this could be work, we can harness our own immune system to attack cancer cells. Weapon 4, what we have is called as an antibody drug conjugate. The difference between weapon 2 or weapon 4 is that here you don't need to have that protein that sits on the surface to cause the cancer. But only thing you need here is that you need to know something that is only present in the cancer cells. Again, we are using the same weapon, antibody, but the difference is you use a payload here. And this helps to target only the cancer cells. And then the payload is basically a toxin. And once it recognizes the cancer cell, it releases this toxin, and then the cancer cells die. That's why it is important that you recognize this weird guy that is only present in the cancer cells and not present in the normal cells, so that the normal cells are not affected and only the cancer cells are affected. Do we know this weird guy that is present in EHE? We don't have enough data to show that what that weird guy is. So in the lack of these information, it looks like the weapon of choice right now, what we have is the small molecule one, which is weapon 1. Well, there is a problem for it. To use weapon 1, there is a prerequisite. And this prerequisite is the weapon 1 has to need a pocket that can to bind. So now are pockets present in TAS-CAMTA1? So basically it needs a pocket so that it can bind. So do you have pocket in the TAS-CAMTA1? This is the monster. As far as I can see, there seems to be no pockets that binds to it. But that is really a naive way of saying that there is no pocket, because frankly speaking, there is not enough research done to show that the pockets actually exist in TAS-CAMTA1. So in the lack of that evidence, basically you like to have a structural data to show that pockets actually exist in TAS-CAMTA1 so that you can use your weapon 1, which is a small molecule. But there is pockets that are present in TEAD so that you can leverage TEAD. Like as Dr. Rubin mentioned, you need TEAD to turn on the genes. It takes 2 to tango. So because there are pockets in TEAD that will prevent TAS-CAMTA1 to interact, you can use TEAD to target the monster chimera. Basically, you're targeting its partner. So this is depicted here. So TEAD has a pocket, and then the genes are turned on when TEAD interacts with TAS-CAMTA1. So basically what you are using here is using as a small molecule to block this pocket. When you block this pocket, the monster is unable to interact with TEAD and the genes are not on. So bottom line is, this is not rocket science, but this is pocket science. So how do we know that TEAD has pockets. So this is something that comes from my earlier study that I did a long time ago. For the first time, I show that you have something called, I've been calling this a central pocket. So you have something called as a central pocket that is amenable to small molecules. So there is a small molecule that can bind to this central pocket. But now we know that actually there are three ways of targeting TEAD. So one, the central pocket is shown here, and on top of it, TEAD has other two pockets shown by the mesh-like diagram here. This is the example of a Novartis compound. This is currently in clinical trial. We do not know exactly what the structure of this compound, and this is kind of, Dr. Rubin calls this like an asteroid diagram. So basically, this is a real TEAD. And what you show here, what I show here is that the pocket, and this small molecule is actually sitting in this pocket. So why is this strategy so successful? I mean, this strategy has been so successful because small molecules are really tested, and it's been in the clinic. And that's why three companies have leveraged the availability of this pocket. I mean, let's not underplay it. Having pockets is kind of a rarity. And mostly 90% of the proteins do not have this pocket. We are fortunate that TEAD has this pocket and we are leveraging it. So two companies, basically Vivace Therapeutics and Ikena Oncology, are targeting this central pocket, basically so that the TAS-CAMTA1 or TC is unable to partner with it and turn on. And Novartis is targeting this surface pocket so both these, actually what it does is that the monster is unable to interact with this partner, and that's why it cannot turn on the genes. So now, okay, well, the companies are doing it. What are you guys doing now? So now we have the mouse model, which came after a lot of effort. And then we have cell lines that we can use to experiment our hypothesis. I mean, we go wrong a lot of times. So it's important to test whatever we have thinking in these kind of models. So even if we go wrong, all we lose is maybe a mice or a cell. So you need another general to carry this task, and that's me. So what we are doing can be broadly classified as three things. First of all, a lot of companies are approaching us with their inhibitors and check whether they work on EHE models, cell lines, mouse models. So we are doing that work. And second, we have our own TEAD inhibitor drug development program. I'll briefly touch on that. And another aspect, which is actually funded by the EHE Foundation with me as a PI, is that we are trying to find other partners of TAS. So basically, this monster, we know one partner that can be targeted, which is TEAD. But I'm sure there are many other partners, and that's the aim of this project, to find other targetable partners. So the aim one, or whatever working, whether TEAD inhibitors work on EHE models. So a lot of these are under NDA, so I cannot disclose, but this data, which Dr. Rubin already showed, is actually a TEAD inhibitor. And what you see here in this plot, when this curve goes down, means that the cancer cells are dying. We actually added TEAD inhibitor to the cancer cells, and the cancer cells are dying. So this is a proof of concept that TEAD inhibitor works on cancer cells. So the second part, I can briefly summarize. So this is a lot of effort to come up with a new small molecule that binds to the pocket. So we know now Vivace and Ikena has taken up this space, and Novartis has taken up this space. Can Cleveland Clinic take up this space? This requires a lot of effort. So the main lead here is Sean Stauffer. So I'm a co-project leader. I help with this because I've been working on TEAD for a long time. So I'm here as a catalyst to accelerate this project. This project is funded by NCI, NIH Next project, and this requires a lot of effort to do something like this. The third thing is about finding TEAD and TC partners. So basically what we are doing is that we're trying to get this guy, the monster Chimera, out of the nucleus, or either that one or TEAD out of the nucleus. And the tool we are using is that small molecules, which are already available. So basically, this is for people who really want to know what we actually do, the details of it. For the other audience, the bottom line is we are using about 4,000 drugs, and we can actually look at TC or TEAD under a microscope, and then we are looking what drug can actually move this away from the nucleus, or what drug actually reduce the level of TEAD or TC in it. And we have pretty much success in it. So that's a talk for another day. So basically what we are doing is that if these drugs actually work, they could be repurposed. So you can use this drug or if everything works, we could probably start a phase 2 trial with the drug that we identify to see if it works. So that's what we have been doing. So none of these can be done in isolation. So this is the phenotypic screen team. Our lab members, Dr. Rubin already showed our lab members. Without a combined effort and the support from these leaders and also from the foundation, this would not have been possible. And we at the Cleveland Clinic truly believe every life deserves world-class care.
Thank you for the presentation and the work. We just have a little time for a question here, and we actually have, I think, a pretty important question coming in from kind of a patient's perspective on this. I mean, first of all, it's just amazing to have gone from, identifying the mechanism of this disease, getting the models, and now actually having drugs that can be tested. I mean, that's the dream, right, for cancer research. The question gets to, if we have these pocket fillers or, any of the 3 areas you talked about, what's your thought on whether these will prevent progression or treat existing disease? Do they have the potential to do both?
I will be hand waving. I mean, basically the 2 of the groups who are actually doing the clinical trial, they're going to present at ASER next week. I don't know what the outcome of the results are. By looking at it, basically I'm drawing inferences from what is already published in there, from the studies in there. It looks like these guys are not killing cancer cells, but they are stopping them from growing. So basically what it is doing is it's stabilizing the disease, and that's what we are seeing. But I don't know if they actually see that in the clinic. But having said that, still stabilizing the disease is still a big deal. You don't have to shrink or kill the cancer cells. I mean, the whole success of BLIVAC is like, for a lot of the cases, the disease have been stabilized. So that is still a boon for the patients.
Oh, it'd be incredible. Yes, absolutely.